Nematodes cause serious damage to ornamentals throughout Florida. Most nematodes are too small to be seen without an aid of a microscope. As a result, you often may not realize they are causing problems, and instead blame plant damage on disease, insects, or nutrient deficiency. I'm Dr. DeBusk, and this video will discuss the biology and type of nematodes, how to determine a problem, and management. Nematodes are unsegmented roundworms, different from earthworms and other familiar worms that are segmented, or in some cases, flattened and slimy. Many kinds of nematodes may be found in the soil of any landscape. Most are beneficial, feeding on bacteria, fungi, and other microscopic organisms. Some nematodes are used as biological controls to help manage important insect pests. Plant parasitic nematodes are nematodes that feed on live plants. All plant parasitic nematodes have a stylet that is similar in structure and function to a hypodermic needle. The stylet is used to puncture plant cells and then inject digestive juices and ingest plant fluids. Most of the plant parasitic nematodes that are important on ornamental plants in Florida feed on roots. Some plant parasitic nematodes, called ectoparasites, remain in the soil and feed by inserting only their stylet into the root. Other nematodes enter the plant with part or all of their bodies and are called endoparasites. Some endoparasites, called migratory endoparasites, continually burrow around inside the root. Other endoparasites, called sedentary endoparasites, establish permanent feeding sites inside the root and remain in one place. As it matures, a sedentary endoparasite's body changes shape, and adult females are usually swollen. Foliar nematodes are important pests in some ornamental nurseries in Florida. Occasionally, these nematodes also damage landscape plants. These endoparasites parasitize the parts of the plant that are above the ground. They feed inside the leaves and buds of ferns, chrysanthemums, strawberries, and many foliage ornamentals. They cause angular spots on leaves that are similar to those caused by bacterial leaf spot pathogens. In landscapes, these are typically problematic only in shady areas with poor air movement. These nematodes can distort or kill buds, distort leaves, or cause yellow to dark brown lesions between major veins of leaves. Pine wilt nematode infests pine trees. These nematodes are spread by certain beetles. The nematodes plug up the vascular system and reduce the flow of water up to the foliage. The pine needles droop, turn brown, and die. Pine wilt nematode is native to the United States, and our native pines are usually tolerant of them. However, in times of drought, pine wilt nematodes will cause additional stress to the trees. Pine species that originate in Asia and Europe, where pine wilt nematodes are not native, are very susceptible to pine wilt nematodes. Susceptible pines include Scotch pine, Austrian pine, Japanese black pine, Mugo pine, and Japanese red pine. Root knot nematodes are the most well-known of the plant parasitic nematodes and the most important nematodes on ornamentals in Florida. These are sedentary endoparasites. These nematodes can damage most woody ornamentals. Root knot nematodes inject hormones into the roots that cause knots or galls to form. These galls are one of the few nematode symptoms that are easily recognized. Root knot nematodes cause extensive physical and chemical changes in the plant that can make it more susceptible to fungal and bacterial pathogens. Some of these plant pathogens cause rotting of the root systems, and others cause vascular wilts. Sometimes the damage caused by the nematodes and other organisms together is worse than that caused by either organism separately. Woody ornamentals that are most often damaged by root knot nematodes include boxwood, butterfly bush, hibiscus, gardenia, bottlebrush, pittosporum, Japanese holly, rose, lantana, and ixora. Root knot nematodes may also harm many popular annual bedding plants such as impatiens, coleus, and salvia, and potted plants. Several genera of nematodes are sedentary endoparasites that occasionally damage ornamental plants in Florida. These nematodes do not cause galls like root knot nematodes. Most typical symptom is an unthrifty root system. Reniform nematodes are semi-endoparasitic, partially inside roots, and limited to soils with high silt content. In Florida, they are found most often in the Miami-Dade County area and parts of the Panhandle. Reniform nematodes are damaging to some ornamental plants and tropical fruits in the limited areas where they occur. In Florida, the citrus nematode is a common problem on citrus, but can also damage olives and persimmons. This nematode normally does not kill trees, but causes them to be unthrifty and have reduced fruit production. This nematode causes a disease known as slow decline of citrus. Lesion nematodes are migratory endoparasites common in Florida soils. These nematodes usually cause dark, sunken lesions on roots. Lesion nematodes are damaging to ferns, bulbs, and citrus, but some species can damage other perennial plants as well. The coffee lesion nematode causes citrus slump, a devastating disease of citrus. Burrowing nematodes are very damaging, but not widespread in Florida. Citrus nursery stock are regularly inspected for this nematode, which is a regulated pest in Florida. Spreading decline is the disease of citrus caused by burrowing nematodes. Burrowing nematodes cause lesions and rotting of roots. In Florida, these nematodes are damaging to citrus, banana and its relatives, palms, and other ornamental plants. Several ectoparasitic nematodes are capable of causing damage to many landscape plants in Florida. The most destructive of these is the sting, awl, and stubby root nematodes. Feeding by these nematodes usually causes a root system to be stunted or stubby looking. Sting nematodes are found in sandy soil and are common throughout much of Florida. All nematodes usually are found in wet habitats such as near ditches, ponds, or poorly drained areas. Several stubby root species are found in Florida, and one or more usually can be found in most Florida habitats. Other ectoparasitic nematodes that parasitize some landscape plants are spiral, ring, stunt, sheath, sheathoid, dagger, and needle nematodes. Not all nematodes are harmful to plants. Some are beneficial because they parasitize pest insects. Others, known as predatory nematodes, feed on other nematodes. They help minimize the number of parasitic nematodes that parasitize desirable plants. Other nematodes feed on bacteria. They may play key roles in the decay and return of nutrients from organic materials to the soil. With any plant problem, an accurate diagnosis is important to address the problem and to avoid wasting effort and making unnecessary pesticide applications. Generally, nematode symptoms are similar to other disorders, so visual inspection is not enough. The only reliable way to determine if plant parasitic nematodes are involved in a plant problem is by having a nematode assay conducted by a professional nematode diagnostic lab. The Florida Nematode Assay Lab at the University of Florida will evaluate nematode samples for a cost of $25 each. See the Florida Nematode Assay Lab website at the address shown here for information on forms, fees, and sample submission. The lab staff will assign a level of risk to the sample based on what kind of nematodes are found, how many of them there are, and the type and age of the plants involved. To make an accurate nematode diagnosis, the lab must have a quality sample. Whether you are submitting a sample to the Florida Assay Lab or another lab, following the guidelines on the following slides can help ensure that the results will be as accurate as possible. Sometimes samples are collected before planting to find out if nematodes are a potential problem, and if so, to take protective measures. Pre-plant samples will typically only consist of soil. After planting, samples are collected from sick plants to determine if nematodes are the cause of plant decline. These samples require both soil and roots. When sampling before planting, collect soil from 8 to 12 locations in the area where you intend to plant. Samples should be taken about 6 to 8 inches deep. If the soil is dry, dig down to where there is some soil moisture to collect the samples, and do not include the dry soil. A small handful of soil from each location is adequate. Combine all the soil into a single plastic bag. The total volume of soil from the samples should be about 1 pint. Samples may be taken with a shovel, trowel, or other device. If using a shovel, you can put part of the soil from 8 to 12 shovels full into a bucket. Thoroughly mix the soil in the bucket and then take out a pint to submit for analysis. Often a nematode assay is needed to determine if nematodes are causing a plant to get sick. For this type of sample, both soil and roots are required. Dig soil and roots from underneath the canopy of the symptomatic plants. Soil should be collected from where most of the fibrous roots are. Usually 6 inches deep is sufficient. Collect roots that are less than 1/4 of an inch in diameter. If multiple plants are affected, collect some soil and roots from several plants. Place the soil and roots together in the same plastic bag. A minimum of 1 pint of soil and 1 to 2 cups of roots are required. The following practices are recommended regarding sampling. Nematode samples should be kept in plastic bags to keep the sample moist. Do not send samples in soil test bags. Soil testing bags are designed to dry soil out. Dried out samples are no good for nematode diagnosis. If using a self-sealing bag, tape this seal shut so that the bag does not come open in the mail. If submitting more than one sample, make sure that the outside of each bag is labeled with a permanent marker. You can also write on masking tape stuck to the bag. Do not put paper towels inside of bags because they decompose and become illegible. Keep samples out of direct sunlight or heat. Heat and ultraviolet light kill nematodes. Even a few minutes on the dashboard or in the back of a pickup can invalidate assay results. Keep the samples in an air-conditioned room until they can be shipped. Handle the sample gently and pack it well. The more the soil gets banged around, the more the nematodes may get destroyed. Fill out the information on the forms with as much information as possible. The diagnostician needs detailed information about the type of plant or plants in question and the cultivar if known. Make sure that the information on the form matches the identification on the sample bag, that is, front yard, backyard, etc. Once you know that nematodes are the problem, you need to manage them. There are limited practices that are effective for nematodes on ornamentals. There are no chemical nematicides labeled for use on landscape plants that have been effective in University of Florida trials. The bionematicide Melicon WG contains a fungus that parasitizes nematode eggs. Results from University of Florida trials indicate that Melicon can suppress but not eliminate nematodes on shallow-rooted perennial plants. When purchasing Melicon, have it delivered directly to you from the manufacturer and keep it frozen until it is used. To apply, mix Melicon with water and then either drench or spray it onto the soil around the base of the plants. Organic amendments can be added to soil as compost, manure, mulch, or other materials. Organic matter can help prevent nematode damage in several ways. The organic matter increases the ability of the soil to hold water and nutrients and to improve soil structure. This makes a better environment for most plants and can help the plants survive in spite of the nematodes. Organic amendments can also increase natural enemies of nematodes that suppress the nematode populations. Some organic amendments can release chemicals or gases that are toxic to the nematodes. There are several organic nematode management products for sale. Researchers with the University of Florida have worked with a number of these, but probably not all of them. In the majority of cases, these products work no better than adding any other less expensive organic material. For more detailed information on these topics, see UF-IFAS publication EMY 59, Soil Organic Matter, Green Manures, and Cover Crops for Nematode Management online at the web address shown here. There are many steps you can take to avoid or at least minimize nematode interference with growing perennial plants. Combine as many of these steps as possible into an integrated nematode management program to get the best chance of managing a nematode problem successfully. Some of these tactics may seem futile or even silly, but each can make a real contribution to suppression of nematode problems. Pest exclusion is the most important strategy to prevent nematode problems. Sometimes plants come from the nursery already infested with nematodes. Avoid introducing nematodes that you do not have on your property already by carefully inspecting plant roots before planting. Do not use plants whose roots have galls, are rotten, or have other abnormalities. Do not plant an ornamental plant that is highly susceptible to one or more kinds of nematodes present on your site. If you must plant an area with high risk of nematodes, consider soil replacement, which requires you to dig out and remove all soil from a generous planting hole, at least 3 feet diameter by 2 feet deep, and replace it with soil free of nematodes and other pests. The plant roots eventually will grow out of the clean soil volume and there will be some movement of nematodes into the clean soil, but the plant will have a jump start on them by having some time to grow nematode free. Soil solarization involves covering soil with clear polyethylene for an extended period in the summer. Solarization of field planting sites in Florida may be disappointing because it rarely heats soil sufficiently at the recommended depth to provide adequate control for the entire root zone area. However, a limited volume of otherwise good topsoil or soil mix can be solarized by completely enclosing it in clear polyethylene film and keeping the soil depth at no more than 6 inches. The soil should be located to receive full sun all day and should be exposed 6 weeks or longer in June and July. This might be a good way to reduce nematodes and fungi problems in existing soil or in a soil mix to be used in soil replacement. In this video, You hopefully learned about the biology and types of nematodes that are problems for ornamentals, how to diagnose the problem by sending in a nematode assay, and how to manage the problem.